description quantitative polymerase chain reaction, also known as RT-qPCR, and it can also be found as QRT-PCR. So what is it? So according to the NIH, it's a huge accomplishment in the realm of PCR technology, and it further allows for accurate detection and measuring of products created during the PCR process. So one workflow is shown on the right here with the TaqMan. Here in the annealing process, the reporter fluoroporin emits a wavelength and is absorbed by the quencher. From here, the DNA polymerase starts extending the primers, moving towards the probe. Then the probe is degraded and the reporter is furthermore released from the quencher and therefore emits the fluorescence. And this fluorescence is how we can see how much, and this is how we measure products of the items created by PCR. So how does it work? I know we went through the workflow, but we're going to go a little bit more in depth here. So usually you'd start by mixing your RNA with the reverse transcriptase and other reagents to synthesize cDNA. From there, you set up the quantitative PCR by combining the cDNA with primers and DNA polymerase in a thermal cycler. And then from there, you would use the machine. It would cycle through various temperatures, and then it furthermore allows the DNA to amplify, and the fluorescent signal increases at a proportional rate to the amount of DNA being produced, or the amount of product being produced. So what do we need to perform the lab? We need RNA samples from cells or tissues. We need reverse transcriptase for cDNA synthesis. We need PCR primers, so we need that for specific targeting to genes. We need DNA polymerase. We need dNTPs to build the building blocks to build DNA. And we can also have buffers as well. What does the information provide? It helps us qualify gene expression levels in various samples. It allows us to identify how genes are regulated under different conditions. It allows for various testings revolving around the realm of pathogen detection, so i.e. COVID tests, RNAi validation, and genetic testing. How do we interpret the data? So the data is placed into our quantitative PCR plot that is shown here on the left-hand side. So we have the baseline, which is shown at the very bottom. And it pretty much displays when fluorescent signal is beneath the limits of a detection, so we can't see anything. We have the delta Rn, so this is the increment of fluorescent signal at each time point. It's also plotted versus the cycled number. We have the threshold, which is a signal detected above the threshold, which is considered a real signal to be used to define the threshold cycle. And we can see CT, which is the basic principle of real-time PCR. It's an essential component in producing accurate and reproducible data. And that is it. Thank you.